Okay.
Um, this is the mycology, and this is dermatophilosis. And then this is our sample collection unit. Our main lab is over there. What we deal with in dermatophilosis division is basically fungi, but we also deal with bacteria. We grow and culture bacteria also, but basically we deal with fungi, that's skin infection of both humans and animals. Now, for us to diagnose our fungal infection, we need our potato dextrose agar, which is known as the PDA. This is used for growing the bacteria. We use it for seeding. That's the technology used. We collect our samples via scraping. That's by scraping the infected part of the skin to get the dry scales from it, that's dead skin already. Then when we get it, we use our wire loop for seeding. We pick part of the scraping and we seed it into the potato dextrose agar for the fungi to grow. When the fungi grow, and it takes it 3 weeks for the fungi to grow on the potato dextrose agar. When it grows, we tease it. We tease, then add methylene blue to it, and then we view under the microscope to ascertain the species of fungi we're dealing with, because fungi are of different species. We have the aspergillosis, we have the trichophyton, we have the mucor, and so many. They are so numerous. So we allow it to grow, after which we now view it, and then our results will be ready. That's for skin scraping. Then when we come to bacteria, we use the BHI, that's blood heart infusion agar. That's what we use for growing our bacteria. Now in bacteria, we don't use scraping, but we use pus. We collect pus from the sample. That's in some of the rashes, you discover that the rashes will form pus. So we prick the rash, then we use our swab stick. This is our swab stick to pick part of the pus. When we pick it, we inoculate into the BHI and we incubate for 24 hours at room temperature, that's 37 degrees Celsius. After 24 hours, we now culture on either blood agar, that's a blood agar plate. That's for bacteria that will need a kind of an enriched media. Blood agar media is an enriched media. But we also use the plain nutrient agar also for growing, for culturing some of them, depending on the kind of bacteria we are suspecting. So after culturing it on the agar plates, we also incubate for another 24 hours. After 24 hours, we run sensitivity tests using our sensitivity discs. Sorry that we didn't have any of our discs here. We use so that we will know the kind of antibiotics that a patient can take. After we've gotten the type of bacteria, then we run the sensitivity test to know the drugs we prescribe for our patients. And we also produce soap, Dermatocide 3M soap, which I don't have one here with me. And we also produce Dermatocide 3M ointment that we give to our patients. It helps cleanse off the rashes, and also it takes away the itches that our patients always complain about, itchy skin or rashes on the body. We use this. Now, the way we use our soap, we apply the soap on our skin, on the body, that's on the affected areas, before taking our bath. For adults, it's for 5 to 10 minutes, while for children, it is for 2 to 3 minutes. After which you take your bath using the same soap. When you're done bathing, you now use our cream. to apply all over the affected areas, parts of the body. You do that daily, after which you come back for your results in 3 weeks, and then we prescribe your oral drugs to be taken. So basically